In this tutorial, we will be discussing the primary structure of proteins. A peptide bond is an amide bond that forms when the COO group, which is the carboxylate ion of the amino acid, reacts with the NH3 group of the next amino acid. The linking of the two or more amino acids by a peptide bond forms a peptide. Peptides formed from 2 aminos are called dipeptides, 3 aminos tripeptides, 4 aminos tetrapeptides. A peptide bond is a amide bond. So here we have the carboxylate ion there and the ammonium ion there. And what's going to happen here is 2 of these hydrogens is going to bond with this oxygen here. And these 3 things coming together is going to make your water. Now what happens to the rest of this is this carbon here and this nitrogen here are going to come together to form that peptide bond, the amide bond. So here we have a peptide bond of glycine and aniline. This oxygen and these 2 hydrogen are going to come together to form the water. Then this carbon and this nitrogen are going to link up to form that peptide bond. With the exception of the C-terminal group, which is right here. This is the N-terminal group because notice that there's nitrogen at the end. This one's the C-terminal group because it's the carbons at the end. And it always goes, if we're reading left to right, the ammonium ion's always on the left, the carboxylate ion's always on the right. Alright, so with the exception of the terminal group, everything else ends in YL, as if it was a substituent. So alanine becomes anilyl, glycine becomes glycyl, and serine stays serine. So we put all those words together and that forms the name of the peptide. Now for simplicity's sake, what they typically do is they just take the abbreviations, ALA-GLY-SER, to abbreviate the peptide. A protein is a polypeptide with 50 or more amino acids that has a biological activity. The primary structure of a protein is the particular sequence of the amino acids held together by peptide bonds. This is important because this is the first level of understanding what's going into a protein and into enzymes. Here's an example. Notice that every single peptide, amino acid, is going into the name. A thyroid hormone that stimulates the release of thyroxine is a tripeptide with the amino acid sequence of GLU, HIS, PRO. Although other amino acid sequences may have these 3 combinations, or these three amino acids in different combinations, it's only when they're structured in this specific combination that will actually trigger the hormone. So where it is in that primary structure is super important because we only have 20 to work with. There's only 20 total amino acids. So arranging them in different orders is essential. And that is the primary structure of proteins.